Okay, great. Well, yeah, Emily, whenever you're ready, go for it.
Okay, yeah, all right. Well, thank you so much for inviting me to speak today. I'm excited to talk about freshwater mussels, any opportunity I get. I am the senior conservation biologist at the Xerces Society for Invertebrate Conservation, who manages our freshwater mussel conservation work. And I just wanted to give a quick overview of Xerces. I know I've worked with a number of you, but for those who aren't familiar, the Xerces Society is a nonprofit dedicated to invertebrate, which includes insects and also other invertebrate species like freshwater mussels. We're dedicated to the conservation of these species and their habitat, and we work on terrestrial and freshwater ecosystems in North America largely. And so we have a freshwater mussel conservation program that focuses on Western North America, and I'm going to talk about some of that work today. But I also wanted to give some background on just freshwater mussels in general, because I'm not sure how familiar folks might be with this species group. There are a number of bivalves that are found in aquatic ecosystems, both marine and freshwater. And so there are some really important life history differences, and it's possible that you've encountered some other species in our freshwaters that are not what we consider freshwater mussels. So just as some quick background, in the John Day Basin and elsewhere in North America, and in fact found on almost every continent now, is an invasive species, which is known as Corbicula fluminea, or the Asian clam. You've probably seen these in the John Day River and tributaries. This is also a bivalve found in freshwater, but it has some distinct differences from our native freshwater mussels, namely in how it reproduces. So it doesn't use a host fish, and I'll talk about that in just a minute and what that means. You may also have heard of zebra or quagga mussels, also called drysenids. These are obviously the really concerning species that we don't want introduced into our Western waterways. Again, a very different species biologically from our native freshwater mussels, in how they reproduce, but also in that they affix themselves to habitat, to structures, and especially infrastructure. So they're a really concerning invasive species for that reason, because they can cause damage to our infrastructure. And they do that by attaching using abyssal thread, which is similar to how our marine freshwater mussels, or excuse me, our marine mussels, blue mussels, will attach to rocks in the intertidal zone. So those are some that you may be familiar with, but our freshwater mussels that are native are really different from other mollusks or other bivalves. So they do use a host fish to complete their life cycle. They use a foot to actually burrow into the bottom of our streams, and so they'll form large beds of mussels in some cases. And they're a really long-lived aquatic macroinvertebrate. So compared to other invertebrates found in streams, they can live on the order of decades to over a century. And they are highly imperiled globally and in North America. So freshwater mussels have several needs that are pretty important to consider, as you think about where you might find them in the landscape or what resources they might be affecting. They do require things like perennial flows. So this is a species very different from the intertidal mussels because they cannot withstand being exposed to air for periods of time. So they do need permanent water sources. That water needs to be clean and provide enough food for them. They need stable habitat features and burrowing material, and that's in part because they have these a muscular foot that they use to burrow into the stream bottom instead of just sticking themselves to substrate. And then they also need this host fish. So I'll talk about that next, because this is really the coolest part about freshwater mussels, in my opinion. And this is what really has made them so successful in freshwater ecosystems. So a freshwater mussel is characterized by this unique relationship with a host fish. So an adult mussel will be burrowed into the bottom of a stream using its foot. The male will release sperm into the water. The female mussel is going to actually filter that out, just as she's filtering to breathe and feed. And she actually has eggs that are located in her gills. And so these are called marsupial gills because that's where she's going to brood those young. When they mature into a larval stage, that larval stage is kind of packaged up and spit out by the mussel. It's full of these little glochidia, which is the name for the larval stage. It looks like a tiny clam. And that glochidium is going to have to attach to the gill or the fin of a mussel, of a fish, in order to complete its life cycle. So it parasitizes the fish for a very short period of time. It'll be attached to that fish for on the order of several days to perhaps several weeks. But when it attaches to the fish, it's actually going to become encysted by the host fish. And so it's really kind of inert other than being attached for that period of time. And that's really beneficial for the freshwater mussel because that's how this mussel is going to be transported from where it was released from the adult mussel to potentially new habitat upstream or downstream. And this is a really important adaptation because freshwater mussels spend most of their lives in the same spot that they dropped off as a juvenile mussel. So this is really their opportunity to adapt to a very different environment than the marine environment. So in the marine environment, those mussels are able to release their offspring into the water and mix around and float around and land somewhere. In the freshwater ecosystem, the river is moving one direction. And so this is an opportunity to move upstream. So very beneficial for mussels to expand their habitat range and to be able to colonize new places. So once the mussel drops off of the host fish, it lands in the substrate and it's going to begin growing and filtering to spend the rest of its life. So there are a number of adaptations that our native Western species have developed to be able to make sure that they encounter a host fish. In the Eastern US, there are some really cool mussels that have developed lures to attract fish to them. In the Western US, our mussels release these packets called conglutinates, or they entangle fish as they swim by with kind of a sticky mass. And that's how the glochidia will encounter the fish. So really unique life cycle that sets them apart from those other aquatic bivalves. So again, mussels, they do require fish-bearing, permanently inundated habitat. They are really cryptic in stream, so it can be hard to identify them if you're not looking for them specifically and have that search image. They can blend in and look like rocks. They're relatively immobile, as I said, spending most of their life in the same place, and they can occur in high density. It's important to keep in mind that they're present year-round in our stream bottoms, just burrowed. And they can be really sensitive to impacts to their habitat and water quality. So we have just a handful of native species in Western North America, so west of the Continental Divide. These include the Western pearl shell or Margaritifera falcata, which has a lifespan documented over 100 years. It requires just one group of fish to be able to reproduce. It's considered a host fish specialist, and that's for salmonids. So you will not find this mussel in places where you don't also find trout or salmon. It has kind of a broader distribution in Western North America and can reach really high densities. We also have several species of floaters, which are, they look similar in appearance and can be very hard to tell specific species, but we do have a handful of species in Western North America. They have generally shorter lifespans of 10 to 20 years compared to Western pearl shell. They are host fish generalists, so can use a wide range of host fish, native and some non-native, although recent research has shown that non-native species tend not to serve as good host fish. They have a wider distribution ranging at one point in time from northern Mexico all the way up to Alaska, depending on the species, and east to Montana and Utah. And then the last is our Western ridge mussel, which I'll talk about more in this presentation. This is a species with a lifespan of 30 to 60 or more years. And we're not sure entirely about the host fish, whether it is a specialist just on sculpins or whether it uses some other small fish for reproduction, but we do know that sculpin are an important host fish for the species. It does have a more limited distribution in Western North America, and Oregon actually comprises really a core of the species range, I would say, in Western North America. And it is a very cryptic species and has really suffered from the greatest declines. And so it's been petitioned for endangered species listing, which I'll talk about in just a minute. So with species identification, you can tell what species it is if you have just the shell of the animal. There are diagnostic features on the inside of the shell, excuse me, that can assist us in determining species. But also there are characteristics on the outside of the mussel's mantle tissue, which is what you see here. These are called papillae, and you can see that our western pearl shell on top, floater in the middle, and western ridge mussel on the bottom have differences in how they look. So these can be used to help without even having to handle the mussel or being able to tell from a live mussel versus a shell. So North America is a biodiversity hotspot for mussels. We just have a handful in the western US, but there are or once were really about 300 species found in North America alone, which is nearly a third of what's found globally. But most of that is east of the Continental Divide. And unfortunately, many of the freshwater mussels in North America are highly imperiled, with more than 90 listed as endangered or threatened. And more than 30 are believed to be extinct now, so more than 10%. And the reasons for this impairment vary depending on the location. So there are historical impacts in the eastern US from things like overharvest. So mussels were once harvested for the pearl button industry, also were harvested for use or collection of pearls. Although they don't make beautiful pearls, they tend to make lumpy, less nice looking pearls. So the ones that are you could buy today are cultured specifically using a different method. There are chronic threats, things like habitat alteration, water management, and introduction of invasive species such as the zebra mussel that have impacted populations. And then there are some recent and emerging issues that I'll talk about a little bit, including what we consider enigmatic declines, mass die-offs, evidence that some populations are no longer reproducing, and then the impacts of climate change. So I'll just mention briefly, Western Ridge mussel is also referred to by a scientific name, Ganidia angulata. The species was petitioned in 2020 because review of its distribution and presence at historic sites, as well as evidence of mass die-offs, indicated that this species is highly imperiled. It received a positive 90-day finding in response to the petition, and now the Fish and Wildlife Service is conducting a species status assessment and will provide a 12-month finding in 2024. So we will know more in a few years as to whether this species is going to be listed or not. But currently we have no listed species on the federal ESA. So I'm going to transition now to talking more specifically about the John Day Basin, and I couldn't do this without first mentioning the work of partners, the Confederated Tribe of the Umatilla Indian Reservation, and I'll refer to that as CTUIR from here on out. But CTUIR has had a freshwater mussel research and restoration project since 2002. It's currently headed by Dr. Alexa Main. This project has embarked on a number of important aspects of freshwater mussel research and monitoring and restoration topics. I'm showing here a map from their recent master plan, which I encourage you to read. Freshwater mussel conservation, supplementation, aquaculture, restoration, and research. The image that you see at the right is an example of the results of multiple years of resurveys of freshwater mussel monitoring sites in the John Day Basin, specifically within the Upper John Day in the ceded territory of the tribe. They have largely worked in the North Fork and Middle Fork John Day, although they also have a number of other survey sites and projects going in other sub-basins in the region, including the Walla Walla and Tucannon. The program is doing incredible work. Most of what we know about host fish for our Western species comes from their lab effort. So I encourage you definitely, if you're not familiar with their work, to reach out and learn more. It's quite active project doing really great work. So want to give a plug into that. I'm going to talk more about my own work in the basin, but want to acknowledge that there's been great work going on for many years. Okay, so talking about some of the work that I've been involved in, one of the major projects that I've been working on since 2020 has been with the Prineville BLM. So I know a couple of you are on the call, and I just want to acknowledge the Prineville BLM staff who have been heavily involved in developing a sampling plan for evaluating the status of freshwater mussels within the Prineville district. So this is a project that was initiated using a random selection of sites throughout the district to assess the presence or absence of freshwater mussels by species across BLM lands. It includes both the Deschutes Basin as well as the John Day. So I'm showing sample sites that were selected from both of those basins here in red. We've conducted many of these surveys already. We still have more planned in the coming years, but as you can see, it's got some good coverage over many of the basins and streams in the John Day. So information from those surveys and also from many of the records for mussel observations in the basin are presented on this slide. This is information that is part of the Western Freshwater Mussel Database. It includes species records from a number of sources. It includes things like eDNA samples that tested positive for freshwater mussels, visual surveys by a number of people, as well as museum specimens. So we looked back at things like the Smithsonian Museum and other collections to look for shells that had been deposited. So this is sort of our more recent understanding of freshwater mussel species distribution in the basin. I will mention, of course, that the tribes in the area would have a much greater knowledge of the distribution within this part of the state. This really represents collections or observations really starting from the 1800s to more recently. So we have an incomplete picture at this point, I would say. But we do have some really interesting findings to date, I would say, as well. We see all three genera of Western freshwater mussels, of lower North American Western freshwater mussels present. So among the floaters, there is a species found only in Alaska. So we don't have that one here, but we do have the Anodonta, the floaters, the Margaritifera, Western pearl shell, and Gonidia, Western ridge mussel. And they can be found individually or in mixed beds of mussels. We know this from, again, eDNA visual surveys and museum specimens. And of course, there have been differences in survey coverage. So there are gaps in places, in part because those have not been heavily surveyed, but also random sampling, like I showed in the previous slide, have helped to fill in some of those gaps. And then there are differences between the subbasins. So I'll pop through this pretty quickly to share some interesting observations. The first is a number of these observations that you see are the results of recent eDNA sampling. I'm only showing positive hits here, not those that were sampled and did not detect mussels. But what you can see is that information is contributing greatly to our understanding of mussel distribution. For example, the presence of floater mussels in the main stem John Day. So this is, and as well as the Western pearl shell observations from some of the tributaries to the major rivers. So very helpful when people are including freshwater mussels in their eDNA sampling efforts. For the Western ridge mussel, we've seen some interesting differences between subbasins. And so, what I'll point out to you here is that despite eDNA testing for Western ridge mussel in the South Fork and Upper John Day, as well as a number of visual surveys by myself and others, we do not find evidence of Western ridge mussel in that subbasin. We do see it in each of the other subbasins. And so just really interesting question of why they wouldn't be present in one of the major subbasins. I also wanted to take a moment to talk about some of the more interesting or noteworthy observations from the lower basin. So if you see the plus sign, that means it's a historic site. If you see the X, it's a recent site. And if you see what looks like a circle, it's where both have been observed. And so just going through this, you know, at Clarno in 1971, someone collected and deposited some shell material in a museum. So we would suspect that Western Ridge mussel would have been present in the lower John Day River at that site in 1971. I personally resurveyed that site in 2018 and only found evidence of shell fragments. So really disappointing not to find a mussel bed still there. But we see similar trends elsewhere throughout the lower John Day. For example, at the Priest Hole site in 2020, Fish and Wildlife Service staff reported finding just a large number of shells, and I'll show a photo of that in another slide. In 2021, Xerces and BLM staff resurveyed that area as well and documented 49 shells as well. And so the concern is that this might be evidence of one of the die-offs that I'll talk about. We have a record from the fossil area in 1952. To my knowledge, that hasn't been resurveyed. It's kind of a vague site, but it would be interesting to know if mussels are still present in that area. And then at two sites in the river just upstream of the Service Creek vicinity, we've seen evidence of a potential die-off there as well, with sick or dying mussels, as well as very rapid declines in the number of mussels present, even between 2 years. In the Kimberly area, again, there were observations reported in 1979, but in 2018, when our team surveyed there, we found only 4 live mussels and many shells. So although Western Ridge mussel is still present in the lower John Day, the trends there are incredibly concerning and warrant more surveys. And so mass mortality events, again, are a major concern. Some of what I'm describing would maybe fall more under the enigmatic decline of they're dwindling in number and we're not sure why, but there's also been recent events in other rivers where we've observed mass mortality, where a large number of mussels will suddenly die over the course of a single season, and in fact, within a month. One example of this that we documented this summer in the Newaukum River in Washington. Someone reported observation of a large number of shells at a site. And our follow-up revealed that in just about a month, we believe that half of the mussel bed had died. And so, you know, by placing quadrats down, we were able to determine that in each square meter, we would detect almost 100 live and almost 100 dead. So these things can happen rapidly, and we don't really understand the causes. And so that's one of the activities that I'm involved in, is trying to better understand what might be contributing to these really sudden mass mortality events. So I've been working with WDFW, ODFW, the Fish and Wildlife Service, and the University of Wisconsin-Madison to better understand what's leading to these sudden events. And just as an example, I'll point out the image on the bottom right is from that Priest Hole site where the 49 shells were observed. So I could go into more detail about this, but in the interest of time, I'll keep moving along. So, you know, I've told you a little bit about what a mussel is and where they're found, but I do want to talk a little bit more about why we should care about freshwater mussels beyond the fact that they are just really fascinating creatures. They are providing so many ecosystem services to our aquatic ecosystems. And I would say that up to this point, really, we haven't appreciated what that means. So freshwater mussels are constantly filtering throughout their lifespan. So they are capturing nutrients. If we think about salmon as bringing nutrients that are marine derived back into our freshwater systems, what we're also seeing is that freshwater mussels are helping to slow that pulse downstream. Mussels will be filtering resources from the water. They'll feed on them, but also whatever they're not actually going to eat, they will kind of package up and spit back out. And that becomes food for grazers. It has led to, you know, measurable increases in other aquatic macroinvertebrates that fish would feed on at a site, for example. We've seen evidence that lamprey grow faster in the presence of freshwater mussel beds. There's also research from the southeastern US that has shown that freshwater mussels, when they co-occur with fish in drying pools, those fish have higher survival. Freshwater mussels feed on things like E. coli and other bacteria, so they could also potentially be benefiting fish by removing disease-causing organisms. So they're food for other species, they create habitat, and they just provide us a lot of resources, including removing pollutants from our drinking water. So many things that mussels do for us are often not measured. And so I really appreciated this example from a recent research paper that showed kind of what happens when we lose a freshwater mussel bed. And so you could imagine this in the case of a freshwater mussel bed undergoing mass mortality. So we lose ecosystem services like biofiltration, nutrient regeneration, nutrient storage, and habitat. And this can occur not just over the short term of hours or days, but really decades, because it can take so long for mussel beds to rebuild after they've been impacted. So it's really important that we conserve freshwater mussel beds where we can, because those ecosystem services do not recover quickly because of the lifespan and nature of mussel reproduction. Okay, so I'm going to jump next to some of the projects and resources that I've been working on or involved in with some of the folks on this call. So the first that I wanted to make you all aware of, if you're not familiar with it, is the Freshwater Mussel Best Management Practices Guidebook called Conserving the Gems of Our Waters. You can access this for free online, but it's guidance essentially for restoration planners, site managers, fish biologists, and contractors to ensure that when a restoration project occurs in stream, that mussels are included in planning for that project, in part because mussels are sensitive to habitat disturbance. So there are measures that can be taken to help avoid impacts to those species when you're doing beneficial in-stream projects. If you have questions about that, I'd also be happy to talk with you more. Another project and resource that is currently in development with the BLM, C2IR, the Fish and Wildlife Service, and the Forest Service is a development of a visual survey protocol framework. The idea behind this is that, lots of people are looking for guidance in how to document the presence of freshwater mussels in the streams that they are surveying. There are a number of recommendations out there, but the goal really is to help solidify and standardize a particular method so that we, as a community, can start collecting freshwater mussel observation data that's comparable and can be analyzed to better understand trends in mussel distribution or abundance. So this is an opportunity for you all to help contribute to better understanding of freshwater mussels in the John Day Basin. We are developing another version. So we have previously developed the first draft of the protocol. We are now undergoing revision and developing a second draft of the protocol. And so that's going to be available for anyone to field test and provide feedback in the coming field season. So if you have interest in trying to collect some mussel data and providing that, please reach out. I'd be happy to connect you with that resource. The idea is that there are varying levels of information that could be collected that would use more or less search effort and would provide more or less information about a particular site or mussel bed. And so if you have an interest, please let me know. Again, this is in development, and we hope to have a published version in 2024 that will be available for people to use moving forward. Okay, Murderers Creek. So this is an exciting project that I've had the good fortune to work on in collaboration with the South Fork John Day Watershed Council and ODFW. This is a habitat restoration project that's taking place at the Philip Schneider Wildlife Refuge, where there's in-stream habitat restoration to improve conditions for steelhead, but there are a large number of freshwater mussels present at the site as well. So we began a project partnership in 2020, when we realized how many mussels were actually present on site. Our early surveys suggested that within the 2.5-mile reach of the site, there were more than 60,000 freshwater mussels present. And so we've conducted surveys to help better understand how many mussels are within the project area. And then we developed and were funded by OWEB to conduct a monitoring project. So as part of this project, we established 51 sites within the 2.5-mile reach, as well as a portion of the stream, Murders Creek upstream from the project area. And within those sites, we pit tagged over, 6,000 mussels were pit tagged, and we have relocated some of them upstream of the project area, as well as to specific locations within the project, with the goal of understanding how the habitat restoration is going to affect freshwater mussels that are present on site or removed. And it's a really exciting project because with pit tag scanning, we're able to relocate these mussels much more easily to better understand, did they live, did they get moved when restoration occurred? Because again, mussels use that foot to burrow into the stream bottom. So in some cases they will be dislodged. So we're able to track the fates of these mussels as the research, as the habitat restoration project commences next year. So we'll have more information in the future on exactly what's happened to our 6,000 mussels out there. But again, a very exciting project that we're hoping will provide us with a lot more information about basic things about freshwater mussel population demographics, as well as response to habitat change. Okay, I know I don't have a lot more time, but I'll switch again to talk a little bit about more generally in the John Day Basin. What are some of the emerging threats that have the potential to impact freshwater mussels? One of the main ones that I wanted to bring up was habitat quality and drought. Freshwater mussels, they require this permanent inundation of their habitat. And so they are really quite sensitive and can be impacted by even short-term loss of water. And so the example that I have here is from the South Fork John Day in 2020. One of the freshwater mussel research technicians was returning to a site that in 2021, this photo was taken, but in 2020, we had conducted a survey of the site and documented a bed of, more than 500 Western pearl shell and floaters. When we returned in 2021 to do density monitoring at the site, what we found was a dry stream bed. And unfortunately, this happened again in 2022. So, one of the important things about conducting these surveys for mussels is that had we not surveyed prior to this drought event, we would not have known that mussels had been present in this habitat. What we're finding is that the more monitoring we're doing, the more we're able to document where these mussel beds are disappearing in real time. So this is really critical to understanding the distribution of mussels as well as their current status. I also want to mention that this is also drives home the point of the habitat quality. So we need complex habitat in our streams because things like pools are really critical to mussels. And just as an example of that, I wanted to show this image on the far left from, it's actually from the Grand Ronde Basin, but this is Rock Creek. And in an otherwise dry stream in late summer, this small pool was home to more than 300 Western pearl shell. So these types of microhabitat refugia are really important, especially in light of future drought. So just something really important for us to consider as we think about where mussels are going to be found in the future. Other concerns about habitat diminishment beyond things like drought. A major concern for freshwater mussels is the loss of stream bank habitat, particularly in places where streams are overly incised or banks are degraded. In habitat where these banks are wetted year-round, those are really important places for freshwater mussels. In Murderers Creek, we often find large concentrations of mussels, not in the stream bottom, but actually tucked along the bank. So when streams become more incised, we're losing mussel habitat as well. And of course, wildfire is a real concern for all aquatic species because of the effects it has on the stream chemistry and the water quality, sedimentation. We've seen examples of areas that once had freshwater mussels be hit with these large fires. Unfortunately, there's not much research into what happens to these mussel beds following extreme fire events. This is actually a site from the Lake Tahoe area where freshwater mussels had been relocated to a stream from a site that had undergone channel enhancement. And so unfortunately, the relocation site potentially was damaged during this fire. Okay, so just, a closing thought that I like to share, because I know I'm often talking to fish biologists or people who are managing land, but, it's worth noting that efforts to manage freshwater mussels, even in the eastern US, where there are many more species and they're highly imperiled, it's really considered secondary to other valuable species groups or resources, but mussels are really integral to our aquatic ecosystems. And so the loss of these species is really powerful. And so it's important that we consider, protecting existing mussel beds and promoting, their continued presence in our streams to ensure that we don't lose those important resources that benefit fish and other aquatic species. So just really worth noting, I would say. And one last thought, I did want to make a quick plug. The Freshwater Mollusk Conservation Society is an international organization that's dedicated not just to freshwater mussels, but also freshwater snails. And this group is meeting in Portland in 2023. It's the first time they'll be meeting in the Western US. They're coming to Portland, and we're very excited that the Pacific Northwest Native Freshwater Mussel Work Group is hosting this meeting. If you're available, I encourage you to come. If you have an interest in freshwater mussels, this is a great opportunity to connect with others and learn a lot more, and a great schedule. So if anyone has any questions about that, I'd be happy to share them. And then I just want to take a moment to thank the many partners, and I'm sure there are many more that I've missed here, but the work that I've described is only the product of a number of people working together. So thank you to everyone who has assisted with this work. All right, and that's it.
Thank you so much, Emily. That's cool. You're sponsored by New Belgium. I hope they provide you with lots of treats. Great. Anybody have any questions for Emily? I know through the chat we had some questions about Alexa's contact info, and Mitch put that in there, so thank you.
Yes, and I know Alexa is looking for people who are interested in collecting things like eDNA samples or partnering on surveys. So definitely reach out to her if you are working specifically in the Middle Fork or North Fork John Day. She's a great resource.
And it looks like in the chat there's some interest in being able to share this presentation with other people. Emily, if you're okay with sending us your presentation, we'd love to share that with the group.
Yeah.
Awesome. All right. Well, since I'm not